Hello everyone, I'm Yu from Hokkaido University, and then my talk is a little bit off topic from the session theme probably, but I hope my study, this is my PhD study anyway, provides some interesting idea or some data to consider archaeology of North for everyone. So this is the contents, and then I skip here. So reconstruction of human migration during the late Pleistocene is very tough because we don't have much direct evidences. However, these topics always excited us to think about the first migrants into the New World. These fundamental and hot topics are origin of migrants, cultural connection between now and then, and their routes and its timing, and how they adapted to the new environment. So I explained some synthesized migration hypotheses here. Until recently, only one peopling scenario was strongly supported and believed. It is called Clovis first model. In the model, first migrants with sophisticated fruited point went across the Bering land bridge and went through the ice-free corridor in between the giant ice sheets. However, last 20 years, other challenging hypotheses came out and have earned more support with scientific evidences. So these two figures show different peopling scenarios created by archaeologists at the top and then geneticists at the bottom. Obviously, there's no firm archaeological evidences to support migration during and before LGM. Although under the circumstance, because of older sites have been found recently from the south of continental ice sheets, coastal route and Atlantic route models were generated as explanation for dating contradictions. These theories requires very careful and precise choices of research materials and data. In addition to the data quality to prove the route, sample quantity is also powerful evidence. It is impossible to examine the whole series in this paper, so I will briefly summarize recent research results. So in the field of genetics, I'm kind of a little bit hesitant to talk about genetics in front of Professor Dr. Adachi, but I will try. And then many scholars have published their outcomes recently. Here are 7 sites with radiocarbon date and human mitochondrial DNA analysis result. Important point is that there are some haplotype diversity even before 10,000 Cal BP in the New World. This circumstance describes the complexity of migration process into the New World. Interestingly, because human remains are more distributed in the West, the coastal migration theory gathers high interest now. So Paisley Cave is in Oregon, and then it's famous of well-preserved human coprolites. Date back to 14,000 Cal BP. This site also has a stone point, which is different from Clovis complex. So this is a western stem point from the Paisley Cave, and then this is a typical Clovis point from the eastern side of the U.S. And then from the mitochondrial DNA, radiocarbon dating, and point typology, coastal migration theory is now very strengthened. And however, although the number of human remain findings and their genetic background data increased, there is still obstacle to stick them together and discuss migrant population. So Northern North America is the most possible gateway during the first migration. But the area only has one early Holocene site with the human remain. It is pointed in Alaska. Therefore, to consider the first stage of peopling of America, we need to rely on the archaeological studies very much, at least now. So lack of archaeological sites older than 12,000 Cal BP along the west coast requires us to focus on interior sites with rigid dates. So archaeological study of Beringia started during the 1930s with excavation of diagnostic wedge-shaped microblade core. Although World War II and Cold War inhibited transcontinental study, Russian and American scholars continue to seek archaeological evidences representing first migrants' material culture. However, even 90 years of effort, there is no North American artifact can be traced back to the Old World except the wedge-shaped microblade core and its technology. Reason of weak typological connection between the two continents is still unknown. Therefore, the microblade assemblage is very important when we consider the migration process. Ubet's method microcore is known as very distinctive in terms of geographical distribution and then timing of northward expansion. These cores are all classified as Ubet's cores. And then as this slide shows, the core can be seen after LGM in West and East Beringia. And older microblade technology was only found from south of about 60 degrees north during the LGM. So therefore, the microblade assemblage in interior Alaska in the New World also important for the Northeast Asia too. So I have to explain a little bit about core technology here. So please be patient. So most common method to conduct the comparative study is using core technotypology. So microcore technology has been descriptively analyzed based on the technomorphological traits on the core itself. So there are two concrete methods can be seen in Alaska. And then on one hand is Ubet method. This technique uses the bifacial blank and remove the entire long margin of the blank for the platform preparation. On the other hand, the campus method at the bottom, this uses a unifacially touched blank flake and then partial platform rejuvenation usually. So these differences are clearly identifiable on the artifact itself and also by the radiocarbon dates. And then this diagram summarizes the chronology of microblade core technology in Eastern Beringia, including Alaska and Canada, from terminal processing to early Holocene. So the core technology tells us how knowledge and the skill of production continued, changed, and vanished. So the primary object of past Alaska microblade studies was to clarify the systematics between Siberia and Alaska through microblade core technotypological analysis. An assemblage containing Ubet's method microcore is only found from Eastern Beringia Tradition 1 layer at the Swan Point site, only one site and one component. And between 14,000 to 13,000 KBP, there were Chindaning and Nenana complex featured by the teardrop shape and the triangular bifacial point up here, and with really scarce microblade evidences. And then the microblade assemblage rehappening, it's called the Denari complex, appeared later that contains different core technology that called campus method. So interpretation of these phenomena is still controversial, and one side regards them as a reflection of gradual technological shift within the same population's local adaptation, and the other side suggests we see the remains of several different migrant waves and occupying that region. So the most well-represented area of early archaeological evidence sequence in Eastern Beringia is in the interior Alaska. And archaeological sites have densely distributed since late Pleistocene here. So as local importance, microblades continue to appear for 13,000 years and widely distributed. The incised osseous points, I took some example here on the right corner, and then both it's found both from Siberia and Alaska. Although these radiocarbon dates and morphology are different, both are regarded as a composite point with microblades. So thus, Alaska microblades are considered as a very similar hunting tool from the old world microblade assemblages. So under this archaeological background, two major problems can be considered. Chronology of Beringia and the Americas, and continuity of local archaeological complexes within interior Alaska. So for example, Ted Gable and others focused on the systematics of the Nenana complex and the Clovis complex, the relationship in between the bifacial points through Beringer. And other times, West and Holmes suggested that the microblade complex with the original migrant, they are representing the original migrant lineage. Against these studies, Weaver mentioned, he said we should focus more on the local diversity than the huge migration theory. And Schott also recently questioned the simple typological classification of complexes that invite cultural continuity problems. So, and then in local level, after well-preserved sites excavated along Tanana River, which continues flowing to the Yukon, reconsideration of former chronology and new definition for the cultural complexes were required. So some scholars now regard the lithic composition variability reflects the site function and seasonality. However, there are only a few detailed lithic studies that can support these discussions. So my study focused on the second problem and sets 3 objectives. The first is considering cultural continuity through lithic materials other than microblade cores. The second is to compare microblade morphology from Eastern Beringer tradition 1 to Northern Archaic tradition, the Middle Holocene tradition, and attempt to see hunting tool change along microcore change through this. The third is to discuss possible reasons of microblade morphological change or stability with other data. This discussion could contribute to consider the cultural continuity and adaptation strategy at the next stage. And my study used microblade samples from 2 archaeological sites. First is called Dry Creek site, which is located along Nenana River Valley and close to town of Healy. It's south of Fairbanks. And the site primal excavation was conducted during the 1970s, and total excavation area was 357 square meter. There are 2 lake glacial components. The first one is Nenana, and second one is Denari. So bifacial point and microblade assemblage. So according to 2011, the latest excavation, the site is dated around 10,700 years ago. So this component 2 is included in left 3 and parasol 1 layer. And then the component contains the typical Denari complex tools on the right-hand side, and such as campus-type microcores, core tablets, burins, and bifacial points. And then a few identifiable fauna remains were excavated, such as Dolbic horn sheep and wapiti, but the preservation was not very well overall. So next site is Swan Point site, which is located on top of the small hill on the Shoal Creek Flat near the Tanana River. The site has excavated after 1994 until 2011. The lowest culture zone 4B, has 3 hearths, Ubet's microcore, burins, spores, bifacial blank, and mammoth ivories, mammoth and horse teeth, and avian remains. So these cultural zones have rich number of microblades, and this study sets sample groups for each zones, and then CZ4B at the bottom is classified as the EBT1, and CZ2 is a Proto-Northern Archaic, and then CZ1B at the top is classified as Northern Archaic. Then as you see, all CZ4B microcores are made, these types are made as a Ubet's method, and the other the Holocene components has the diverse microcore types, including the semi-conical, also the campus type, and some cylindrical cores. So there's a little, even they use the microblade, but they use a different technology. So this graph shows the total and sample numbers of microblades. It's not cores, only the microblades from the both sites. So the AMS dates on the graph shows the range of the youngest and oldest for each component. I sampled 1179 out of 2201 from the swan point, and it's more than the 60% of total numbers at each cultural zones. And also from the Dry Creek, 1128 out of 1772 sampled from the collection. It is also more than 60%. So because excavated microblades are usually intentionally broken, and so I have to set the complete proximal, medial, distal as segment types at first. And the measurement was conducted for the 4 different attributes: maximum length, maximum thickness, maximum width, and angle going through the line consists of lines that go along platform and also ventral side. Then I conducted the statistical test to compare each sample groups. And another analysis, I used the 95% confidence interval to compare the different groups divided by the different raw materials to see the physical property difference that affects to the size difference. So basically, this is a methodology of I conducted. And then this slide shows the relative frequency of microblade segments and raw material, and the proximal and the medial segments dominates the whole sample usually. And then for the raw material, calcine, rhyolites are common in all groups, but I could see a little bit of unique raw material only seen in the CZ4B. It's called green igneous, but I don't know exact name of the mineral because we cannot identify it through the mineralogy yet. So I just quickly move on to the result. And then hereafter I colored specific groups on the graph to emphasize if they have obvious differences. So when we look at the histogram of platform ventral angle, which represents the technological difference of detachment of microblade from the core. So CZ4B distribution peak is around 70 degrees. It's the left-hand side. But the others have a peak at around 80 degrees. So, and then statistical difference are confirmed between the oldest component from the swan point to the other groups. And then this is the maximum width. And then all sample groups concentrate around the 3 to 5 millimeter range and decrease at 6 millimeter. And then the cultural zone 1B at swan point and then Dry Creek groups disperse more than the others. The maximum width statistical test result shows the difference between Dry Creek group, a Dry Creek sample, and all swan point groups. And in the maximum thickness, only cultural zone 4B at swan point has peak at 2 millimeter, and then the other group have it at the 1 millimeter range. So, and then statistical test result, differences were found between combination of CZ4B and the other groups, and also CZ1B and the Dry Creek component 2. But in short summary, the result of angle may reflect the difference of production technology. The size differences can be interpreted as both site or chronological cause, at least now. And then this slide shows the raw material comparison result. Because of sample size shortage at some groups, and I describe the result as in form of 95 confidence interval. And when we look at the maximum width on the left graph, there are several gaps between Dry Creek and swan point whole groups. And statistical tests also detected the difference in between the sites. And the maximum thickness on the right graph doesn't show any gap within the component or single cultural zone. So statistical difference and gaps can be seen among thicker cultural zone 4B groups and then thinner dry creek component 2 groups, and the other groups. So in summary, raw material physical property doesn't really affect on any size differences. So here I discuss and consider the result of the both size and statistical tests. Because classical Wallace test detects very small morphological differences. Significant point is to consider that the difference reflects the intentional production design or production method by the human being. So the table on the slide shows mean and standard deviation value of maximum width. And swan point groups are less than a millimeter at mean and 2 millimeter at the standard deviation. Dry creek component 2 group mean is not less than about 1.5 millimeter from the swan point. So as an additional data to discuss this issue, I refer to the experimental data and actual ductile Siberian microblade width data. And then this is experimental data produced by the different method. It's a handheld pressure flaking. This is the shoulder crutch. And this is using the berry to have more pressure onto the core. So the handheld, the graph A and shoulder crutch graph B has almost the same distribution peak, although the maximum value is different. So graph D, the ductile microblade concentrates in 6 to 8 millimeter range. So what I can read is that each group shows 2 to 3 millimeter high frequency range before and behind of the peak. So these distribution range is also overlap each other. So according to these data, in context of lithic production, it is reasonable to interpret that the microblade size of this study is relatively consistent. So assuming that the size is very consistent, I need to consider the possible reasons of its stability with other archaeological and environmental data. So environmental and mammal composition data indicated by Guthrie shows the huge shift in late glacial period in this area. So from this data, environmental shift from cold dry steppe to warmer wet taiga is predictable. This shift could happen with faunal distribution change too. So, and then Potter focused on microblades and the mammal remain association using almost all available data in this area. So his study shows the microblade components more associated with large mammals such as bison, wapiti, and moose than only the point components. So his another analysis shows the microblade components tend to form in the lowland environment. So I consider that the microblades were used differently from the projectile points and used as specific hunting tool for the large mammal prey. Then I conclude here. Based on this study, the microblade size in interior Alaska was relatively consistent through the time. On the contrary, a microblade detachment angle shifted as same as microcore production methods. Therefore, my methodological change was evident. So although huge environmental shift and faunal composition shift happened in the late glacial period, microblade and the composite point might be used for large mammal hunting from the late Pleistocene to middle Holocene. This specialized usage is one of the possible reasons of microblade size consistency. But however, identification of microblade size difference is a still problem to be solved. I believe well-designed and specific experimental work for Alaskan microblade will give us a hint to solve this problem. Because microblades were found almost all regions in Alaska during Holocene time, the same concept I used here could be applicable for the later periods. In terms of discussion of human migration and adaptation through technological and hunting tool tradition, these topics are my future objectives. So I got many help from these people who work in Alaska, the museums and the institutions, so I thank for them here. Then thank you very much for listening.